Hi, let's continue on the transport phenomena in biological systems. So today we want to discuss about the relative importance between diffusion and convection depending on the length scales. A lot of these materials are from transport phenomena in biological systems by Trotsky. So I brought this table about relative length scales in biological systems. So we know a lot of these entities, such as proteins and nucleic acids, that length scale is about, I like to put it as a 10 to the minus like n of meter. So that's, we know that the size of the protein is relatively about 10 nanometer order, and organelles are about 10 times higher. And like the cells composing our body for eukaryotic tissue is about, about 10 micron length scale, so which is 10 to the minus fifth length. So that's about 10 micron. And interestingly, the capillary, which is the business end of our oxygen and nutrient and waste exchange at the tissue, is about 10 to the minus fourth meter. And that turns out to be smaller than about 100 micrometer. And so that has an implication today we will discuss. And organ size for human is, let's say, about like 10 centimeter. If it's a kind of big organ, then that's a 10 to the minus first. And our whole body is an order of meter scale. So if you look at the our an organismic body to the smallest machinery, protein, it's like covering a huge 8 orders of magnitudes. And that length scale is an enormous layers of coverage. So what that means is no single transport processes can function effectively over these many length scales. So what we want to discuss here is at shorter distance, yes, diffusion can be quite rapid, while a longer distance, convection typically transport molecules across. So that's we want to argue this time. And what we previously discussed a lot about diffusion and random walks, this is a kind of a hypothetical mathematical description of how we can analyze diffusion process more analytically and easily. So the conclusion from there is diffusion time increases as the square of distance. So what that means is, hey, diffusion time, if the length scale is, let's say, twice, so we want to diffuse twice, then the time for diffusion will be 4 times. If the length scale is, let's say, 10 times longer, and the time for diffusion is simply 10 square, which is 100 times. So as the length scale or the diffusing distance gets bigger and bigger, the diffusion alone becomes a pretty inefficient process. So here I brought this table from the Trotsky book, and here comes some table of diffusion coefficients. So mass diffusion. So for example, here small molecules and gases, those have about 10 to the minus fifth here centimeter square per second, while a proteins inside the liquid has about 10 to the minus seventh order centimeter square per second. So that's something you want to keep in mind. And so we are discussing diffusion versus convection. So in French physicists called Jean-Claude Eugene Peclet already kind of defined this ratio. Likewise, we defined previously Reynolds number, which is an inertial force versus viscous force. So likewise, Peclet defined as a convection, mass transport by convection versus mass transport by diffusion. And it's pretty simple. And you can see here, it's so that's mass transport probably is inversely by the time scale. So it becomes a diffusion time divided by convection time. And how can you define diffusion time? Basically, diffusion coefficient unit. If you look at the unit, it's length square over time. So that will be the time will be length scale divided by diffusion coefficient like this. And then how can you come up with a convection time? So convection is a simple displacement by velocity or mass flow. So that's pretty much the time for convection will be distance divided by characteristic velocity. So that's L over V. And you calculate this and it turns out velocity times length over diffusion coefficient. So what this means is here L is a characteristic length and V is this local flow velocity and D is a diffusion coefficient or mass diffusion coefficient. So if Peclet number is a very big, that means this is a convection dominant, okay? And on the other hand, it's very small, then we can consider diffusion dominant process. So here's the kind of summation, summary for that relative importance of convection and diffusion. So this lower slope is a, talking about convection, while higher slope is discussing about convection, diffusion process. So with respect to different two diffusion coefficient, there's like two crossing over. So crossover happens around, let's say, gases in liquids around 100 micrometer, while proteins in liquids, it's around 10 micrometer and this order. But it depends on how we set up, assume like the characteristic velocity. But the important aspects from this kind of analysis is for proteins, diffusion is efficient, dependent of the size of cells or smaller, which means, you know, like inside a bacterium or a cell, proteins can be very efficiently diffused so that it can cause all this metabolic activity, enzyme activity inside a cell. But for small molecules such as gases and glucose, and diffusion is efficient over the distance of like about 10 times bigger or 100 micron. So that means is these gases can diffuse at least tens of micrometer. So so we have I found a paper in PNAS that what's the normal spacing between capillaries in many tissues. What this reference tells us is generally to less than 50 or 100 micron is capillary spacing. So that means, you know, inside the tissue, which require this gas, oxygen and CO2, you know, like, or this exchange will be efficient enough with the diffusion so that, you know, our convection at the end will be based on this capillary. So the capillary to capillary, the distance will not be bigger than 50 or 100 micron. So that gives us how the nature, when we develop our system for effective convective transport of molecules, and if at the end, out of this capillary, you know, that shorter distance, diffusion is enough, or inside the cell, the diffusion process is fast and efficient enough for this or exchange of molecules.